Thank you very much. Good morning. Thank you all present. My name is Nancy, and I will present the research assessment, namely the assessing the functionality of ectopic leg in spiders exhibiting a Hox knockdown phenotype. So Hox genes are a class of homologs genes in the animals who determine the position of body parts. This meaning they are responsible for determining the general body plan, such as the numbers of body segments, the number of placement of the appendages, and the animal head-tail directionality. Here in the picture, we can see a schematic draw of a fly. The line is ballon and the squares are the representation where each gene is positioned on the chromosome, and the figure indicates where each gene is expressed. In this study, we worked with a gene called antero-posterior. The antero-posterior is a Hox gene that in insects controls the formation of legs during development. However, antero-posterior gene in the spiders play a different role. A study with Parastatoda tepidarium showed that antero-posterior repress legs in the first segment of opisthosomal. To determine that Kajet and collaborators studied the function of antero-posterior by RNAi interference. RNAi interference is a natural phenomenon by which RNA messenger is silenced, thereby inhibiting the protein coded from the particular RNA messenger. There are some macromolecules involved in the RNA interference mechanism, but you can focus in the information that RNA silence is triggered by double-stranded RNA. So the results of Kajab and the collaborators only the morphology of the RNA embryos and the poster embryos was reported, but the functionality of the ectopic legs of the pedicel remain unknown. Then this is the goal of this study. We replicated the experiments of Kajab in collaborators where we silence antero-posterior using maternal RNA interference and thus generating 10-leg spiders. The experiment consisted in 4 females being injected 4 times with double-stranded RNA. They were injected in 1-day interval to recover, and the females were mated after the last injection with untreated males. I'm going to talk now about the results of this study. So after mating, the females produced several cocoons, about 1 per week. The cocoons were collected, analyzed, and the phenotypes were recorded. In this study, the Hox knockdown phenotype could be observed from C3 to C5. Here, image of from ventral, this big one, and the dorsal of the Hox knockdown phenotype in posterior embryo. At the first molt, in the posterior embryo stage, we observed that the molt can be stuck at the ectopic leg, and sometimes the ectopic leg do the autotomy if the spider needs it to be free from the mold. Here another picture of the spiders with the 5 pair of legs. At the second molt of post-embryo stage, we observed that several spiders had their leg regressed. Looking like the leg starts to dry and die. Here, for example, it's just the remainder of the costal segment. And here, some of the segments, here we still have some of the segments, but the leg is drying. And I'm kind this process as the dying leg. The innervation and internal morphology was assessed during embryogenesis. We deployed the fluorescent immunohistochemical staining to mark the tropomyosin. It's a muscle mark, and they were present in the 5 leg. And the acetylated tubulin, it's a neural marker. They were also present in the leg 5. And to assess the proper morphogenesis of the appendages, we assigned for the incidence of proper podomer boundaries and the sensory organs of the post-embryo stage. So comparing the leg 1 and the ectopic leg 5, we confirmed that all podomers are present. About the sensory organs, at the post-embryo stage, they are also present in the ectopic leg. They are these dark dots. And we also use the fluorescent confocal microscope to assess muscles at the post-embryo stages. I'm gonna share with you some of these amazing images. So here we have the 10-leg spiders. Observe the same pattern of muscles in each of 10 legs. Here in the tail, we have the 5 leg and the pedicel. Here the leg with the process I'm kind of calling the dying leg. We have the segments, but the others are dry and dying, and looks like dying. Here in the tail, that leg, and what I'm told him, it looks like he's drying again. So in conclusion, our data show that the ectopic leg pair induced by antenna-PG1 knockdown is functional in a subset of phenotypes. And our next steps now is to discover how we can keep the ectopic leg exhibiting the Hox knockdown phenotype. So I want to thank you very much, the funding agency, the universities, all the presents here, the organization of the Congress. Thanks to the laboratory teams in Brazil and here in the US. They are amazing laboratories where each one is very careful, collaborative, and they are very friendly. And thank you very much, Prasha, for accepting the challenge to teach me this wonderful word of Ivo Devo. And I wanna wish my son a really happy birthday because today is his birthday. He's doing 80 years old. And thank you very much.